that you said that you can't wait for climate change. I'm like, what?
No. I thought you said time change, so I was replying to that. My bad. Yeah, I think so. Yeah. It is what it is. There's not much we can do about it now.
Oh, we're probably not gonna die from it. A lot of people actually can see it.
Not us, but it might be for other people.
Yeah.
Yeah, I haven't updated the numbers yet from the last time, so I apologize for that. But it's just been you guys and my late class that I've had time to do the questions with. My Tuesday morning, I just didn't have the time. So it's fine. I still update them because they're not related to each other really, but it helps you kind of see like how to do in the other copies. Oh. So I warned you guys, chapter 6 was going to be heavy, and it was. So a lot of people go that way anyway. If it was easy for you, I'm happy for you, but for a lot of people, that's a lot. So remember what we learned in chapter 6. We're about to do our Kahoot to review. There's going to be replication, transcription, translation. Those are our big concepts, right? And then how these things happen. Watch the videos. It helps make more sense of it than what I can draw out. So I can recommend at some point watching those. Don't put it off for too long, so it stays fresh in your mind. Remember that replication is just making copies of the DNA. We're doing anything from DNA to RNA to protein. Those are tiny codes. That's one that people miss a lot on the test. I'll take it. All right. So we're going to get started here in a second. Be sure you're signed into this if you want to join. If you don't, I'm not going to hold it over your head, but I do feel like that these quizzes are pretty helpful in reminding us what we learned in the last chapter before we get started with the next one. So make use of it. Also remember that these are available on modules. So if you want to go back later on and take it at your own pace, you can do it that way and help use it to study, whatever you need. So just a couple more minutes and then we'll get going. Anybody have any questions before we get started? I always want to ask that. Okay. Think of any movie now.
Yeah, she's very. Right, I mean, she, whenever I went to the lab, but she already had her little feet, so, you know, the content that this. I was trying. Oh, I know, I'm not. She's perfect.
Well, and Sarah, you know how I am. It's like, I warned that person that if you want to sit by yourself, you can, but you'll be doing all of the work by yourself. You won't have anybody to work with you. And she chose to do that. And if she's not asking for help, and she'll just like have her paper filled out and leave. And I'm like, I'm pretty sure you didn't do anything. Like, but
Walk around and
yeah, right.
by the time I look up, everybody's gone.
Yeah. So like I said, people make their own choices.
Yeah.
They want to dig their own grave, let them. So,
Someone's doing like no work and just sitting there or something?
I mean, she does some work. It's just not really all of what she's supposed to be doing, if that makes sense. She doesn't go to the different groups. I've only seen like one of her slides. She doesn't ask for help for that either, so
I made you extra smart.
maybe, I don't know, man. All right, so first question, what is the term that refers to an organism's total sum of genetic material? Exactly, good job, guys. So that's the genome. Remember, the genome will include like your chromosome and maybe any extra other genes that came in on plasmids. If you're bacteria, you can have those circular DNA with extra genes, or like your mitochondria have their own. So that's still part of the genome. All righty, next question. The physical expression of the genotype. You guys remember this? What is the word for what's expressed? All right, so good job. So this is in fact phenotype. It's pretty straightforward. We only had to learn really those terms of genotype and phenotype. Genes is like saying there is a gene that codes for your hair color. We didn't learn about what alleles are in this course, but if you were to learn about genetics properly, alleles refer to the different versions of the genes. So like the brown hair and the blonde hair or red hair and all of those things that would fall into, those are alleles, different versions of that gene type. But we don't need to know it. But so that doesn't seem like something like you should have known. There it is. Okay, the 3 parts to a DNA nucleotide are good job, guys. So looking for here for DNA, our nucleotides, we know what all nucleotides are going to be the sugar, the phosphate, and a nitrogenous base. All of them are that. But which sugar is it going to be? For DNA, it's deoxyribose. For RNA, it's going to be ribose. And remember, with RNA, it uses uracil instead of thymine as the nitrogenous base. But otherwise, everything's pretty much the same. I'm sure there's questions about that, but that's okay. More review isn't bad. All right, DNA. Okay, what is that about? DNA, anti-parallel strands of nucleotides are linked by. So we're talking about the whole strand and the other strand going the other way. How are they linked together? Right, so this is hydrogen bonds. I'm glad you guys read into that. It's just worded weird.
But now I was like, maybe that's too easy.
Yeah. I tell people, and I'm sure you guys looking, and I'm not, like I said, I'm not trying to be rude about it. It's just how you guys think about it and how you will approach the questions a lot of times, that it's like overthinking it from like the wrong angle almost. So yes. Covalent bonded, those are going to be the nucleotides. Like take what, so we have in DNA 2 strands, right? And they run anti-parallel to one another, right? And we learned about how that ladder, the links here between the ladder are held together by hydrogen bonds between the G and C. So those are hydrogen bonds holding those anti-parallel strands of nucleotides. That's what they're made of, to one another.
Oh wait, is that the strand itself that are?
Yes, so that's what I was going to get at next. It's like each of those would be a nucleotide, right? Nucleotide, making up a strand of nucleotides. So yes, the nucleotide to nucleotide in that strand is covalent bond. So yes, that's why I was saying this is kind of worded weird, because yeah. I can see how we're thinking about that, but yeah. Anyways, I'm hoping that if we can get more exposure to the material and become more confident in it, that when we get to the test questions and we start overthinking it, hopefully we'll have more information to work with so we don't get lost in words. And that's what my goal is for you guys. But all right, true or false, DNA polymerase builds DNA in the 5' to 3' direction. I won't be saying it enough, man. It'll just yeah, so it's always 5' to 3', so good job. If you don't even necessarily need to know it, I'm not gonna have pictures where you have to pick out why it means that or anything. You can remember 5' to 3', you should be good on that. So which enzyme connects the Okazaki fragments formed during the DNA replication? So we've made the actual fragments. How do we make it into one, connected strand instead of like strand that is like got pieces in it? So we only mentioned this briefly, but it is a good one to know. This is DNA ligase. So this is a term to know that will come up in definitely this next chapter. It'll come up a little bit more. So ligase, DNA ligase, it's ligating. If you don't know what ligation is, it just means sealing things together, bonding together. So that's all that means. And it's not trying to be a crazy word or anything, but in case you don't know, that's what it is. So just seals those together. We make the fragments and then turn them into one by creating those covalent bonds between the fragments. So during transcription, what's going to happen? Just to be clear, this is an image of transcription. And I know I had the thing up on the board. You're welcome to cheat from it. All righty. So during transcription, we are going from DNA to RNA. Here in that picture that we saw, I wish it would have kept it up, but it's separating those DNA strands and making, mRNA basically from that gene, adding the RNA nucleotides, creating the copy. And that's what transcription is. Remember, the enzyme that does that is called RNA polymerase. Just like DNA has DNA polymerase. All right, a codon is 3 bases of what? All right, so it's going to be mRNA, the codon. So we have the 3 nucleotides of the DNA called the triplet. The 3 nucleotides in the mRNA that's going to actually call in the specific amino acids from being carried by the tRNA. Those 3 nucleotides on the mRNA are called a codon. The tRNA that's going to recognize the complementary sequence on the codon. So tRNAs, remember, they're T-shaped, right? And at the bottom of their T, they've got a matching sequence to our codon on the mRNA. So remember, A to U and G to C. So they'll match up that way. It is literally complementary. It's still RNA, base-to-base matching. So mRNA codon, tRNA, anticodon. Okay, so that's the terms for those guys involved in all of this. Okay, so that'll bring in the tRNA that carries the respective amino acid that matches that sequence. And that's how we get the correct amino acids when we're reading our genetic code. Okay. Just breaking it down to the parts now and assigning those terms. All right, here we go. In blank operons, the repressor proteins bind to the operators unless a molecule that changes the repressor's shape is present. So if I were to look at this complicated explanation of it, it's saying that there's a repressor there and something has to come along to remove it. Think of that and which term would apply to that. All right, so our 2 terms for operons will be inducible or repressible. And in order to be inducible, you have to be off. And if you have something blocking your way on your operator with your repressor, that's the off position for both of them, no matter what you're doing. If it's there, it's off. This is one situation where we have it off. Something's going to come along, change its shape, like a little clothespin is what it looked like in the little picture. And then it comes off of there so that RNA polymerase can do its job. It's not being blocked anymore. So this is inducible. We can turn it on. Okay. Gotta love operons, y'all. All right, missense mutations result in what? Like I said, it was a lot last chapter, so we're like barely touching on this stuff. Just so you guys know, you guys are missing out on the fun, and so will spring semester. But starting in the fall, they're requiring biology to take this course, both biology and chemistry. Y'all, we've been fighting for so long for this. This has been a fight that science department has been fighting for so long. And finally, so people can come in here, I'm sure you guys can see it now. Obviously, it would help to know things about what a cell is and what DNA is and how it's going to work outside of just chemistry. Yeah. Yeah, so that way they'll have both of those working under that, because you know you need both.
But we don't need it
now.
now.
No, you guys don't. You guys get to suffer. You guys get to suffer without that background. Yes, what's up? Or I mean, if it's helping you, the knowledge from high school biology is helping you with the course now, then it's fine. But they're going to actually require it as like you have to have college level biology. And believe you me when I tell you, college biology is drastically different from high school biology. Drastically. Like when I went in, somebody who was, we would consider a, upper level scholar. I was high achiever. I overachieved, if you will. That like I was only required to have 129 hours to get my bachelor's degree. I graduated with 149 hours. And that's me being overachiever, right? So put that into perspective when I tell you that I went from my high school and then went into college. And one of the first classes I took was biology, thinking I was good at everything, whatever. And it hit me hard. Like I was like, oh shit, I know nothing. I thought I understood science. And it's just like, it's like this stuff. It's talking about ribosomes. It's going to be talking about, we went over stuff about the metabolism that we'll do in chapter 10. But all of that, it's like the basics of that stuff, taking like of this information of these kind of basics and putting it into a course so that when you get to here, it's just reminder. So you guys remember how like when you took when we did chapter 2 and it was the chemistry stuff, it really was a lot of reminder. It was reminder you heard of covalent bonds and stuff like that. Imagine that every chapter in the first part was like that. Imagine that. So that's what it's like. So that's what I'm saying where you guys kind of missed out. Like, yeah, I mean, you don't have to go back and have that credit. But they're going to account for that credit hour change in the nursing program. They're kicking out like something else to make room for it, like a Comp 2 or something like that. But yeah.
In AP Bio, I went over a lot of this stuff, but it's been a while since I was in AP Bio.
Yeah, AP Bio covers the same stuff, right? Because it's college. It's college biology. I'm not going to lie. To be clear with you guys, when I was in college, I enrolled in AP Bio. And when I realized it was new stuff I was going to have to do work in, I dropped that class. I took AP Chem and I just aced that. But like, it was so much new, it was so much new.
Yeah.
I mean, I don't know. So anyway, so like I said, we worked so hard to get this into place. I'm sorry that you guys didn't get the benefit of that before you got here. And so it's going to be harder for you. However, for the future generations, that doesn't mean they get all the same stuff. It means I'm going to break off a lot of this stuff and we're going to be focusing on the harder stuff a lot more. So it's not really a win if you look at it. It just cuts out some of the basics. So anyways, good job on this. That's our Kahoot for the day. And we don't have sound apparently. Oh, there it goes. Just quiet. Like the little sounds. All right, so, whew. So that's what I say to everybody, right? Yeah. Yeah, that's it. So you understand what I went through when I was in school. Almost all of my classes were like that. You were lucky if you had a class that had slides. Like really, like they all wrote on the board and you're like, okay, what is he talking about with blah, blah, blah. And you barely get a chance to even ask a question. If you do, they'll just be like, it's here on the board. Stuff like that. And you're like, okay, guess I'll go back to the book later. Yeah.
So we had like a 5-minute break between, and
Come back and like cover and stuff. Also, AP biology helped me significantly with all of this. I was able to get a biology class at this school for the AP bio.
Yeah.
Plus, we went into even more detail about DNA replication and transcription
Yeah.
and translation. Whenever you mention the wheels, I'm like, that sounds familiar. I'm like, oh yeah, I did
Yeah.
that in AP bio.
It'll lean into things a little bit differently. Yeah.
So, because nowadays, you have like. Is there a way to go back through McGraw-Hill? I mean, so is there a way to go back to where you something?
Yes. So
Being the book that you've already purchased.
if you go into McGraw-Hill, that link on the left side, if there, I don't know what it looks like to you guys. So let me bring it up on my screen and see if you guys can verify that this is what it looks like to you guys, because I don't know.
So much fun.
Oh my God, with the dual push. All right. Okay, so over here on my left side, you guys know this McGraw-Hill Connect. It's the same link kind of deal in my class as it is to anybody else that has McGraw-Hill. It's just we don't have assigned stuff in our McGraw-Hill. I have like practice stuff for lab if you guys want to use it. And it says practice because it is practice. It's not required. You're welcome to do it. It acts out the labs. It's just like any of the other online lab simulations. So it acts it out. So if you want to get reminded of how to do the thing, it's kind of a good reminder for that, but it's there. So you can click on go to my Connect section for that course. All right, and then this is what I see. So, oh, it does have student view. Let's look at this. It'll let me. Okay, so course materials, you can choose to purchase an actual paper copy if you wanted to. It's not going to be included in what you've paid for. Down here, it says read. If you go to read, you can click on that top one. That's our actual textbook. And it'll bring you into the textbook. This is just where I last looked somewhere in the textbook to find like an image or something to put in my slides or whatever. But you can go to the table of contents and actually find like the chapter and go that way if you want to, big time. Or if you wanted to, if you were looking up, let's say operons, right? You can just type in operon here and it'll give you options. So from the table of contents versus chapter 6. So if you're like, yeah, we learned this in chapter 6, you can find it there and find the term that you're looking for more explanation on that. Just breakdown of operons as far as protein synthesis goes. So you could click on, oh, inducible operons, what are those? And then find that there. If you wanted to be like, oh, but what was the information before this? You can click this button up here and it takes you to the previous pages. And so whatever. So each of these sections is kind of a clip. That makes sense. So if you guys haven't used this as a resource for looking things up, it might have better wording or more explanation that makes more sense to you. I can, only cover so much in a certain way here. It also has videos on there that you can get to. I've been told by one of my other students in one of my other sections that there are some really good videos that they've made that can explain some of the concepts pretty well. I just don't have time to play them in class when I have to cover that material. So I try to post the ones that I feel like you really need to see, like the genetics ones. Like I just can't draw it appropriately. So I highly recommend seeing those. But if you want to go see other videos, you can click on some of these little things that pop up. Sometimes they'll have videos that you can go to, like watch video, bacterial transformation, right? So you can go find stuff like that and play it. Cool.
So I think my question, so I know how to get to this, but
Sure.
like say, like previously, our anatomy course, how you have the anatomy book. And so like in here, you talk about some.
Because technically you
physiology.
paid for that
Right,
book.
and you talk some about physiology and stuff, and you're like, oh my gosh, like if you wanted to refresh your memory. Is there a way to go back and look at the text?
That's an interesting question because I always have the access, right? So it's different. I don't know for sure, but I feel like on McGraw-Hill, you should be able to like go to that like main page on McGraw-Hill, not on Canvas, and be able to see past stuff
Oh.
on McGraw-Hill. Yeah, so it's like, if I were to look at like, not this textbook, but like this stuff, you should be able to, I don't know where it is for students, but find other classes that you're in on McGraw-Hill, and it should bring up, yeah, classes, whatever. I just have these, right? And it should have archive classes that you should be able to access the stuff. If not,
Yes.
okay, good. I was like, if not, we can contact McGraw-Hill and get that for you. It shouldn't be a problem because you paid for it. You should be able to keep that. Okay, because I can access all the former editions and everything. So I feel like you guys should be able to also. So good questions. I hope that helps you guys with some resources. Okay, so let's get into genetic engineering. We're going to talk about recombinant DNA technology. Remember, recombinant is anytime you're going to have organism DNA with organism DNA. They can be different species, different things all together, or like just a cell that this strain has antibiotic resistance and it's getting from that strain, and now it has become a recombinant. You're getting it from another organism at all. So using this to our advantage and hijacking the concept of this in nature, we're going to use that to create recombinants of our own. Right? So utilizing DNA to identify somebody in a crime, for example, or to recognize like, paternity tests, things like that. Fixing underlying genetic mutations to treat diseases. I'm about to get to the very end of this one. Hopefully we'll make it through. But at the very end of this one, we'll talk about changing your genetics while you're already alive right now. So if you have a genetic defect where you can't make a protein or something, introducing a gene that works into your body so that it can compensate for that. So we're going to talk about that. That's called, gene therapy is what they call that. All right, and then utilizing CRISPR technology to repair genetic mutations in a heritable way. That means passed down to your offspring. So if you have, historically had this disease in your family pop up every now and then, and you get tested and you find out that you have it. Let's say it's even, the breast cancer gene, the BRCA gene. And you find out you get tested for that, and you find out you have it, and you can do what you need to do for you in that time, but how do you keep your children from having to deal with this ever again? Using CRISPR technology to repair any of those genetic mutations for any future offspring that you could have. So we're talking about like in your eggs or future generations of sperm, depending on what the gene is. So a heritable way, which is, great. I think it's one of the greatest things that we can think of as far as using this stuff in medicine. But of course, it always brings forth that question of where you draw the line. Huh? Yes. So nobody would probably, ask that question for deadly diseases that are causing you problems, or even things that are going to cause like deformities. You would live otherwise. Things that cause disabilities, like that. You might not ask that question. But what about the fact that if you do this, you can also change your eye color. You can also change your height. You can also change the hair color. You can also change the voice. Make your child an athlete. Make your child a genius. Make your child both. So this is called eugenics, if you haven't heard that term, choosing the genes for your future children. And if you haven't seen Gattaca, it's a movie that basically, the story of what spawns from that. And it's like a child that was born naturally to parents, and then a child that they had designed. And then kind of the struggle between them, because the child that they designed was like this cop, and then natural child wanted to go to space, and he didn't think it was fair that, naturally born people couldn't go to space. So he faked that he was, genetically superior. He stole somebody else's skin cells and would sprinkle them around for testing. He would steal he had the person's blood samples. They would supply blood samples to him. And it was somebody who had gone through actual like physical damage, became paralyzed. And so he couldn't go to space anymore. And so he was helping this guy out, taking his identity so that they could make up for that. So it's Jude Law is the paralyzed guy, and Ethan Hawke is the normal born guy. And it's a pretty good movie, back when Ethan Hawke used to be a hero. Gattaca, Gattaca. So it's like a play off of the ATCG of DNA. So I highly recommend. It's old, right? It's an older movie, but I think it still, checks out. So personally, it gives a good insight into like, is it really okay to do this? I mean, what about people that can't afford to have their kids upgraded? And is that really fair to them? And should they be given the same opportunities? And how do we keep that in control? So anyways, that's the big brain stuff behind all this. But for the little brain stuff, we're going to stick with things like making insulin for people, or maybe possibly helping with genetic diseases that are definitely, detrimental. So we already know about DNA and how it's a double helix, and how, like we said in that other question, those two sides of DNA are held together by hydrogen bonds. So, not surprisingly, like we said with pH, if hydrogen bonds depend on that H plus and O negative interaction, and we're introducing a bunch of H pluses, that could interfere pretty seriously with how this is going to work. Same thing with heat. Heat can also break these bonds in a similar way. So putting in that energy of heat, breaking the hydrogen bond. And we have utilized that concept for our advantage in a lot of this. So if you guys have ever heard of PCR, and I know you have, right? Because COVID tells you, you have the home test and you have the more accurate PCR test. We're going to talk about what it is and how it works, but it relies on that concept there. The fact that you can heat up DNA and separate the strands. Okay. And then cool them down and they'll stick back together based on the ATCG binding. Just back to normal, because that's how they're going to do, because chemistry says. Okay, so first thing that we're going to learn about, restriction endonucleases. Nice long word. These are enzyme ACE. Remember, ACE always going to mean enzyme. So endonuclease, think about that as inside the nucleotide area. So inside of your genetic code, we're going to be cutting the DNA. And it's going to be restricted based on the sequence. So that's why it's called this. So normally bacteria make this to act on and digest up like foreign DNA that they don't want in the cell. It's like they're a defense mechanism against foreign DNA. They break the phosphodiester bonds. It's just the covalent bonds between the nucleotides. Okay. Between the phosphate group from one nucleotide to the sugar of the other nucleotide. That's the bond that they'll break. It's a covalent bond. These guys break those, and each type of endonuclease that we know that exists so far recognizes a specific sequence in DNA of 4 to 10 base pairs. So when I'm telling you base pairs, if this strand reads ATCG, that's a base pair. Okay, so it's talking about both strands all at once. So that would be 4 base pairs. This is 4 base pairs right there, or 5 base pairs altogether. Just say that so it's not confusing. That's 5 base pairs. So it's talking about that in terms of like the coding strand and the template strand together. If you remember those terms. All right, so these guys are going to normally protect against any foreign DNA. We said that already. And they're going to usually clip at sequences in the DNA that are palindromes. And yes, I am talking about palindromes like usually, like we talk about in English. You don't know what palindrome is? It's anything that reads the same forwards as it does backwards. So like race car. You guys have never written out race car. It's the same forward and backwards. So we're talking about palindromes and DNA sequences. So it's always that. I don't know why, but it just is. So don't ask the question, okay? And it's just how they recognize things and how it just happens to be. And typically we're talking about palindromes from the actual strand itself as well as on its opposite. So it's pretty cool, and I'll show you what I mean by that. But
I think there was a typo with 5 to 3 direction on one strand and 5 to 3 on the other.
yes, that's correct.
That's correct.
Mhm.
Oh, no, mind.
So remember, if this were to say race car, we'll just leave it at that for now because we can understand race car. But this says race car this way. If it's a palindromic sequence, it's also going to say race car this way. And it would match up. That's the idea of how this works, okay? I'll show you what I mean. So it's still 5' to 3' and 5' to 3'. So if we cut between the first R and A on this one, like let's say here, it would do the same thing on this one, okay? So when we separate it, with our heat, you've got sticky ends. So you'll have this piece sticking out on its own and this piece sticking out on its own. This is highly useful in genetic engineering. You can imagine, because if you are trying to put human insulin into a bacterial plasmid that you're going to put into bacteria, to make that insulin, you can genetically engineer your human insulin with sticky ends that also match in the plasmids, sticky ends. So you can treat them both with that enzyme, sticky end on your plasmid and on your plasmid, and the same thing for on your gene and on your gene, and then they can stick them together and insert. Make sense? So there's a picture I'm going to show you that makes more sense. Like I said, this is one of those things that I can only explain it so much. But we call them sticky ends, and they're staggered symmetrical cuts with a tail. So here's kind of a better picture of it. A few different ones. These are EcoR1. This came from E. coli, restriction enzyme #1 that they found on it. Doesn't matter. You don't have to get tested on that, but that's why they're named what they are. Haemophilus influenzae, but whatever. So these guys cut on palindromic sequences. So yes, it says GAA TTC. But if we have GAA, just looking at that top strand, okay, TTC, what happens if you fold this over onto itself? If you fold it directly over, palindrome. So this is what I'm talking about with palindrome on one strand versus obviously on the other strand, it's like the actual palindromic effect. So if you go GAATTC on the top, the other one is GAATTC on the bottom. And they're both going 5' to 3'. See? So that's what we mean by palindrome. I'm not going to make you guys like draw the sequence and have you guys pick the parts where it's supposed to be or anything like that, or like, is this a palindrome or not? But that is what it's talking about in sequences, what it's referring to, so that you understand what I'm talking about going forward. Okay, so we create these little sticky ends, because if we separate these two strands, now this side, this whole side here, it looks like a dick, and I didn't mean for it to, but it is what it is. That side versus the other side here, those guys will come apart. And in the plasmid, which you would see, this is what's going on down here, right? When you're heating that up after you've cut, cut those ends. In the plasmid, now we have those sticky ends, and then you can insert your DNA from whatever organism, and it has the same sticky end. And you might be like, how do you control the plasmids though from like not just sticking back to each other instead of like taking in the new gene? You can't really like literally control that, but if I put in enough of that new gene overwhelming the population of plasmid in there, it's probably going to stick. So you get at least some. And the plasmids we can design, which we'll learn when we learn about transformation in the lab, but the plasmids we can design to tell us basically if it got the right genes in it, as well as if it is the correct plasmid containing the right genes into your DNA, in your bacteria that you're going to put it into. Okay, so that's the idea of how those work. So then we can create, believe it or not, your genes, because it has to work that way, right? Your genes also have palindromic sequences in them, just randomly, just because genetic code, right? There's just so much variation in it. So you can cut my genes with EcoR1, like we saw over there, and cut your genes with EcoR1. And the fragments that you get should be different, right? Because our genes are just so, like our genetic material is just so drastically different. So those are restriction fragment length polymorphisms. I always have to think it's, so no worries, but restriction fragment length polymorphism. So the polymorphism means there's different versions of it, okay? And these will just allow us to look at cutting sites within certain organisms and work with that, or to identify between people if we're looking at that, which we'll get to in a moment. But those different lengths of fragments that we get from just using those enzymes that we just talked about can be useful in a lot of different ways. That's what this is saying. So there's different size fragments. That's the restriction length fragment length polymorphisms. Jesus, man. Okay. At least you guys don't have to remember, like, write it out. So, geez, oh, okay. Ligase. We talked about DNA ligase, right? And how if you're going to create those sticky ends and you want to insert your gene in there, that's fine, but you can't leave these open. They're not going to create that covalent bond on their own and reseal themselves. That's a problem. So when we're creating copies of our DNA, we have an enzyme called DNA ligase that seals together our Okazaki fragments, right? And that's what it's for. So we'll use that same enzyme and introduce it during our procedure to help seal together our sticky ends. So they're actually going to be useful. just like won't be able to replicate whenever the bacteria try to replicate. And so a lot of this stuff, remember, we're going to be putting them usually into a plasmid, a little bit of DNA that we're going to put into bacteria, and the bacteria have to grow then. And if they grow on their own, that's fine. But if they can't replicate the plasmid, it's useless. So we have to make sure they can actually replicate that plasmid. That's why we'll do it. Okay. All right. So that's typically what we use it for is plasmids. We also have the tool of reverse transcriptase, which we learned about in viruses. Reverse transcriptase lets you go from RNA to DNA. Why would that be important? If you have an RNA, sample from somebody for any reason, whether it's because of a crime scene, you were able to isolate RNA, or you specifically want to get the mRNA out of somebody's gene that's already, remember, already been spliced out those introns and put the exons together. We can isolate that already spliced mRNA to make a copy from it. Because the bacteria can't do anything with that. Bacteria cannot cut out introns, and all of our genes have introns. So if we can get our mRNA that already had the introns cut out, then we can turn it back into DNA and pop that into our plasmid. So reverse transcriptase changed the game whenever it came to that. It's really cool stuff. RNA to DNA. So it goes backwards from what we are used to. So when we do that, when we make, after we have our silly little intron removal, so remember, useless here. Cut it out, cut out the useless via splicing. Now we put together the exons, the useful, to create a working mRNA that our ribosomes would be able to read. So you can isolate that from someone's cell samples and turn that mRNA, which is single-stranded, into a single strand of complementary DNA or cloned DNA, and then make it double-stranded. So we can do that to get the DNA that's properly already spliced so that the bacteria don't have to worry about it, right? All right, next we have CRISPR. I know most of you have probably heard of CRISPR. If you haven't, this is revolutionary as far as science goes. It's not new. People think it's brand new, and it is not. It's been around since the 90s, but we just haven't figured out how to make it useful. And we're working on making it useful gradually, more and more and more as science improves. But this is referring to clustered, regularly interspaced, short palindromic repeats, okay? That's what CRISPR stands for. And we have a system, it actually works in a system, CRISPR-Cas9. Cas9 is the enzyme. And it's going to be dealing with, it's in a gene sequence of CRISPR. It's a whole gene sequence dealing with the whole thing. So Cas9 is the one that's going to be cutting out any foreign DNA from invading bacteriophages. Same concept as the restriction endonucleases, except we can use this to target any sequence at all, ever. So now we're not limited to those palindromic BS. And it just dictates what we can cut at and what we can't cut at, right? That's what the palindromes did for us. Now we can literally choose the sequence where we want to cut in your genome right now. And then hopefully the idea is we can cut it there and then insert a working gene if it's not working. Or cut it there so we can get it out of somebody who's healthy and then create a mechanism for you to get like a virus or something that will put the new gene in, which is the more common way to look at it. But it's revolutionary scientifically. It has not been successfully used in any application in humans whatsoever by anybody anywhere. So that's the problem with it, getting it to work. It's so right now, it requires more knowledge of the genome than we have. And that's the problem. To create a CRISPR-Cas9 system that could cut at any potential version of genes of the specific gene, like this person has this disease, how many genes are involved in that disease? Because a lot of times it'll be more than one. And how to cut them and insert them and keep it safe. So that's the problem with it, getting that stuff worked out. And it's not safe right now. It's not like it's killing anybody. There are people that do this stuff in their garage and they'll swear that it's working, but I promise you it's not actually permanently working. Okay. So PCR, number one, most important thing you're going to learn from this chapter as a healthcare professional by far is PCR. It is so heavily used in diagnostics, you can't afford not to understand this. Okay. So PCR stands for polymerase. You already know what polymerase is. Learn about DNA polymerase. chain reactions. We're having a chain reaction event using DNA polymerase. So guess what it's going to be doing? Making copies of DNA. That's all it is. It's just making copies of DNA. Well, I'm going to show you what that means for us. So sensitive. The test is so sensitive, it can detect cancer from a single cell and even diagnose an infection from one gene copy. That means one piece of DNA, and it can diagnose your infection. That is mind-blowing. There's home tests ain't doing that, right? You need to have enough to cause that little thing to change the color. Not PCR. One virus. You can detect one virus if you get it in the sample, you know? So that's the idea. That's why they're saying it's more sensitive than the home test, because y'all, it is more sensitive. Guess why, though, they don't do it unless you really need it done now? It's more expensive too. And obviously it takes longer than the home tests do, but usually not prohibitively longer if you're diagnosing in a hospital setting. It's usually 1 to 3 hours to get the answer. So you're not waiting days, usually. Waiting just a few hours to get an answer. So you might be thinking that's the end all be all. You don't need any other test. But I'm going to tell you that you're wrong in a moment. So first of all, PCR relies on the events of DNA replication. We're going to open the double helix and use each strand as a template. So open it up and remember that semi-conservative replication. So we use the old ones as the template, make the copy on each side, and now you have 2. In each round of PCR, you're going from 1 strand to 2 strand. So if you have, that means, so then if you, in the next round, because you have 2 strands, each strand is going 1 strand to 2 strand. So you're doubling each round, which can take you up to billions after like 40 rounds, which is nothing in PCR world. That's like you waiting the 2 hours. So it's very impressive. So we're talking about from 1 copy into billions. You're just like having your coffee break. You can see how that's useful for diagnostics, because if there was even 1 copy of that thing there, and you can't have it no other way. You have no other way besides PCR to see if there was 1 thing there. But you can run this and it'll tell you if it was there. No other way to know it. In just a few hours, you can know it. So that's useful in and of itself, but it's also useful in genetic engineering, which you can imagine. When I told you we have to flood the area with the gene in order to get it to, bind into your plasmid, you can buy plasmids and cut it with your stupid enzymes. That's fine. But you can't buy your human insulin gene. Scientists have to make that. So somebody had to make the plasmid too, but come on. So anyways, you have to make that. So you can make a few copies of that with your enzymes and with whatever DNA you extracted from human cells and whatever, get that ready. And you only need a little bit of it, put it into your PCR machine, and now you've got a lot of it. And now you can put it in with that plasmid and you're almost guaranteed to make it how you wanted it. So that's what this is useful for. You can see the uses that could be coming out of this as far as both diagnostics as well as genetic engineering. Massive, massive benefit to the medical as well as the scientific world. Yeah, and a lot of times medicine benefits from the scientific world, typically. So cool, so cool. We'll use each of those exposed strands as a template, and then we'll add our primers in, because remember, DNA polymerase doesn't add off of nothing. It's like trying to jump a chasm as a rock climber, and you can't reach the other side. Right? You're not going to do it. It's like bringing that side of the chasm closer. So adding in something in between so you can get going. Okay. So it has something you can build off of. You have to have something to get going. And that's our primers. The primers also serve a different role here. Not just that, like what we see with DNA replication, that's what we see, that's all it does. But with this, you can design your primers. So this way, I'm not copying your entire genome, just popping it into PCR and hoping for the best. This way, I'm targeting a specific gene. So I can have like 30 nucleotide primers instead of just 4 or something. And they will only bind into specific areas where that sequence matches, the A to T and the G to C. If they can't find that sequence, that means the COVID DNA isn't there. And that means it's not going to multiply. It wouldn't need to because it's not there, right? So if they were negative, that's what I'm saying. So if your primers can't bind to something and work off of your primers that you designed for the COVID genes that you're looking for, then you're negative. And that's how we know. Whether you're gonna polymerize or not is really the answer for PCR. But that should get you guys thinking about, wow, that's pretty cool. You can just design a primer to, you know, identify the disease and whatever. But that should get you guys thinking about how now for every disease that we know of, you would have to have primers for it. Because one primer don't work for every disease. And having primers designed for these things, it doesn't just come like all the time. Otherwise, some of these diseases that we don't use PCR for, we would be using it. It's either hard to find a gene that is reliable to diagnose that disease. Maybe some versions of salmonella don't have that gene. We can't find one that they all have that would work reliably and that sort of stuff. That can be hard. And all sorts of stuff. Not to mention, if you go to the hospital right now and you had like some obscure disease like Crimean Congo hemorrhagic fever virus, like I used to study. Let's say you had that disease. You go to BACT and you're like, okay, I have these symptoms. And they're like, well, we think it's CCHFV. But are they going to have the primers for that? No. So they will take a sample from my blood and probably send it to the CDC where they likely do have the primers. So you'd have to think about stuff like that. So now we're waiting days, not a few hours. So sometimes it's easier, especially with bacteria, to identify them with what media they grow on or what, you know, metabolism they have going on, like we're going to be learning in the lab, which they do this actively, like for some diseases, they prefer to do that. Not to mention when I was in my food lab, we ran, a lot of companies ran on PCR. Many of them were fine. If it came up positive or negative, they were like, yeah, good, whatever, that's fine. Most of them, if they popped positive, wanted us to confirm that it was positive. And it wasn't just some contaminant from our lab. We're dealing with samples, you know, every day. E. coli, Listeria, Salmonella were the common ones that we did. So they'd be like, well, you say that we have Listeria in our sample, prove it. So then we would go through all of those biochemical tests that we're learning in lab. Stains, we do gram stains, we do motility, we would do special media, that was selective media. We would do catalase tests, which we'll learn at the end. And there was some other stuff that we did in the middle. Like 7 steps just to confirm what the PCR told us. So that kind of puts it into perspective how sometimes your results may not be as reliable as you think they are. If you're working in a clinic where you're handling heavy samples of people who are probably COVID positive, and that happened, probably that's what was going on in the beginning. What is the likelihood that somebody's going to be mishandling and getting your samples contaminated with positive? It's pretty nasty high, probably. So you might be misdiagnosed as positive when you didn't have it. It's less likely now because we don't have so many people coming in getting tested, but that can be an issue, especially in an outbreak. So think about that too. If it's that sensitive that it only picks up on one piece of DNA, y'all, it only picks up on one piece of DNA. That's all you have to get contamination wise. You guys probably can imagine how much contamination you probably already get on your samples in lab that you just can't see. So that puts it in perspective, I think. So useful, yes, but sometimes you need something to verify. So as far as this, how this stuff works, I definitely want to cover PCR pretty well. If we're going to cover anything well, I really want it to be this. We do our primers that are going to help us define a specific gene, and we have our DNA polymerase. And what we're going to be doing is heating up our DNA sample super, super hot, really hot. Like, I think it's 92 Fahrenheit, 92 Celsius or something. What does it say? 94 degrees Celsius. 94 degrees Celsius is, you know, near boiling point. So 94 degrees Celsius, that hot. Now, most of our proteins, even our own DNA polymerase, aren't going to survive that heat. They would be broken down too, not just our DNA. So we have special DNA polymerase that we have isolated from one of those thermal vent bacteria called Thermus aquaticus. And it lives on those thermal vents where it's like way over boiling temperature. And we isolated its DNA polymerase and we use that. Revolutionary as well. This is what allows this to happen. So it gets super hot, separates the strands. Next step, we're going to cool it a little bit to allow our excess amount of primers to stick onto their sequences if it matches. Then we're going to be adding on nucleotides in the 5' to 3' direction. Ding, that's it. That's all. That's one cycle. And then you just do it over and over again. Like literally that is all PCR is. 3 steps over and over and over again. So denaturing is separating the strand, priming, adding our primers on, and then extension, adding on our nucleotides. That's one cycle, do it again. All right. On the exam, I don't know if it's on, pretty sure it's on the lab exam. You will be asked if you go through 4, start with 1 piece of DNA and go through 4 rounds, how many pieces will you have? And if you guys need to like draw it out, that's fine. I'm not judging because you'll get scratch paper. So, but think about that. That's something if you're going from 1 to 2, 2 to 4, it's the second round, 4 to 8 is the third round. So 8 to 16 is the fourth, is this, yeah, 4th round. Yeah. 16.
So we're doubling
Every time, every single time it's just doubling what you had in the previous one. Yeah, so it's really not that complicated when you break it down like this, and especially looking at it in the picture, you're just redoing those steps over and over and over again. So we can get that. Additionally, those nucleotides that we're adding on the green strand as it's building longer and longer in the picture, they can be attached to fluorescent molecules. So as they're being added, they'll kick off the fluorescent molecule and fluoresce, and the machine can detect it. So it will know if it's adding on A, if it's adding on C or T or G. They'll all have their own color based on what they're using. So you can buy them like that. And the certain machines can detect that. So it can basically tell you, yeah, we're building this sequence out as you're going. And at the bare minimum, it can tell you we are successfully adding on nucleotides every round and actually building something with your primers that you gave us, as opposed to not building anything if it didn't match. Right? That's called real-time PCR. So you can literally see the progress real time. Most diagnostic facilities, including the food place that I worked at, use real-time PCR. So you get a sense of whether it's positive or negative within like 20 minutes, maybe. So it's pretty useful. Neat stuff. Science, man, right? All right, so we can use this stuff for recombinant technology to form genetic clones. Cloning is removing genes from an animal or something and putting it into another organism. So we have the donor and we have the, cloning host, if you will. Usually we're going to introduce that into some sort of vector like a plasmid like we've been talking about. It could be a virus as well, which is pretty common if we're doing this to actual people. So anyways, we'll put it into those vectors to insert it into the host, which is going to become the clone. So genetic donor, like let's say it's the human pancreas, that's the donor. The vector, we're going to put it into a plasmid, the gene for the insulin, and then insert that into the bacteria, which is our cloning host, and then create clones by growing them up. That's cloning. Okay, that's all it is. It's not always making a sheep. Okay. Isolating genes. What am I trying to say about this? You can amplify them from PCR. You can cut them with endonucleases, and you can use reverse transcriptases. We kind of already talked about all that stuff. Our vectors, we've already introduced the plasmid. It's a double-stranded DNA circle, and it usually has other things like antibiotic resistance and stuff on it. I'm not going to go over the specifics of how that works in your favor right now, because we're going to talk about it in the lab. I'm more concerned about it then. But it'll have, resistance for antibiotics and certain places that can be cut with certain kinds of enzymes. So you know that you're using the right ones that'll actually match with your plasmid and that stuff. And then we can also use bacteriophages to insert DNA. This is what a plasmid would look like. This is our map of a plasmid. This is PUC19. So the P is stands for plasmid. This is what a plasmid would look like. It would be ampicillin resistance. So that's what that means. You're thinking that's what it looked like, then you were right. And then we have a zone here for maybe lactose usage. Why would that be important? Well, you see how all of this BS over here, like EcoR1, that should look familiar, right? That's all of the possible restriction enzymes you could use to cut this. And if you want to be sure that you cut this and that you are actually putting it into something that had, the gene that you're interested in, you can interfere with that gene by getting it into the middle of that gene. So if it can use lactose And now we're breaking it up so it can't use lactose. That gives you information on whether you have the gene or not. That sort of stuff. Whether it made it in there. So we'll have it usually in places like that. And then other indicators or selections like ampicillin resistance. So now you can take your bacteria, your usually E. coli, plate it onto media containing ampicillin. If it has the plasmid, it can grow because it can resist the ampicillin. If it doesn't have the plasmid, it's not going to grow. So that's how you can select for the bacteria that actually took up the plasmid there. So they have the plasmid. Do they have the gene in there? Well, you know, basically how it reacts with that stuff up there will tell you if it has the gene. Or based on the production of the gene itself can tell you if it has the gene, right? Like we would be doing in our lab, we would be inserting GFP, which is the green fluorescence protein. And if it gets inserted, if you get the right plasmid with the right gene inserted in there, it should glow green. Hey, it's not going to do that naturally, right? So now you know that you got the plasmid with the gene in there and not just the plasmid by itself. So that's how it works. We can use this to produce certain things like drugs that we will treat diseases like interferon, which is just like you get biologics like Humira or Remicade or any of those that are like blah, 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 MAB, right? If it's MAB, that stands for monoclonal antibody. It's a specific antibody, and you can put that gene into bacteria and have them spit out that antibody as long as they have that specific gene. So now you can crank out an antibody against whatever. In the case of Humira, it's an antibody against the receptor for one of our inflammatory genes. So we send a signal in our body for inflammation, and it reacts to its receptor to tell the cell to do the thing for inflammation, right? We can make an antibody that binds there so that the thing can't send the signal anymore. Yes.
Why is it so expensive to obtain?
Because you still have to make it. So that's the problem. It's not like it's easy. Can you make a plasmid? To build DNA from scratch is not easy and it's not cheap. You have to be able to make nucleotides and things like this. And then we're talking about getting the resources for making them and making sure, that's the other thing, right? Making sure they're 100% pure. Because when I worked at the transplant lab that I worked at, when we handled people's DNA samples, so we get their blood and all this, and we would spin it down, we get their plasma so that we can select their specific, their white cells and whatever. And then we would go deal with it. After we did that, go deal with it in our clean room. And so we would have to walk into the clean room. We had to walk through the sticky thing. And then we would put on like slippers and like special lab coats that were only for in that room. And we had to clean that room every single day from top to bottom, basically with bleach and everything to remove any possibility that there was any other DNA that could contaminate your patient's samples. So that sort of stuff. So if you are making nucleotides or making primers or making bits of DNA, you need to be concerned about that sort of stuff. And ordering stuff from companies that are going to make that stuff for you, you're asking them to do that in a clean environment too. So then they have to have people that are trained to do that. They have to have the facilities to deal with it. They have to have HEPA filters probably and all that stuff. So you're paying for a lot more than just, nucleotides. So that's the problem. Not to mention, like if you want to create a drug for blocking, inflammation, you have to do the research to figure it out and like which gene are we going to be blocking or which molecule are we blocking, and then design it first before you can start making it en masse. So that takes a long time as well. And then you have to, pay for all that.
Does that like kind of get into, I'm trying to like probably like uncharted water because it's like saying that this cost is more than the supply. It's something that's needed for a person, but the cost associated with going about it, it can be, sometimes like death.
Why don't you ask the government that? So yeah, I mean, we could all argue that, so that gets even into the whole ethics of universal healthcare, honestly, right? But who pays for that? You do. So does it count really that you're saving money? So if you have to pay more money for their treatment, that's why America's not keen on it. So it's not an argument to have with me because I'll just be honest, I'm pro universal healthcare.
Well, I was asking.
Yeah.
It falls into the charted water system.
I mean, it doesn't in the American government. Literally, it's like cancer, right? So If you had cancer that was caused by smoking, let's say, you smoked when you were 18 years old and you get lung cancer when you're 48 years old and you're gonna die, you have to pay for everything out of pocket. Your insurance will not cover it. And this isn't a problem that other countries have.
Wait, what?
Yeah, because you caused the cancer. Your insurance company will absolutely prove it to you that you caused it.
What?
Yep, I'm not kidding, guys. You don't know that.
If someone else gets cancer from your smoking?
No, if you get cancer because you smoked when you're 18,
Oh.
if they have any evidence whatsoever that indicates that you smoked ever in your life, then they'll send you one of those little letters that say, hey, can you call us? Because we have a discrepancy here. We're not paying until, and then if you say, no, I didn't smoke, and they say, oh, well, look, it looks like you did.
But that you did. Let's say I smoked at 18 and I stopped when I was 18.
It wouldn't matter.
That would have been based like one time. Just out of curiosity.
They'd have to prove it, right? So they'd have to prove it. That's the thing. But like there are diseases that are like that. Maybe not lung cancer, where we can get in this argument of like, what if I only, oh man, if I only smoked that one time or whatever. We can get into those arguments. But there's diseases that are like that right now in the world that you live in here in the United States. That you would say it isn't your fault though, but your insurance company and the United States of America say that it is. And so you'll have to pay completely out of pocket for it. We're talking 50 grand, 100 grand. Yeah, right. More than that, depending on the disease, right? So, yeah, and it's like, let's say, okay, here's the best example I have. Diabetes. You get type 2 diabetes. Super common. If they can show that you were overweight up until that point, they ain't paying shit. They have the right not to, because they have the right, because it's linked, it's been shown that being overweight is linked to having type 2 diabetes. If your doctor told you that you needed to get on a diet and lose weight at any point before you got type 2 diabetes, the insurance company doesn't have to pay out. Yeah. Yep, it's the same thing. Right. If it's type 1, it's different because you're born with it. If it's type 2, and like we were saying over here, Ozempic, if you guys have type 2 diabetes and you want to go on Ozempic, which is originally used to treat diabetes, right? So even though it's going to treat your type 2 diabetes and bring you down to a normal weight where you no longer have diabetes, they won't pay for it because you did it to yourself. You pay for it. Well, if you can prove that, it isn't your fault, if it's genetically related, yeah. Yeah, it just depends on the situation. And they'll look at it situation to situation, but they, in case you guys haven't noticed, they always lean towards not having to pay.
Yeah.
So, yeah. And then what happens when you go to the ER and you have insurance and you're there for a reason that was like legitimate, and you're still paying thousands of dollars with insurance, or you're out of network. Exactly. What happens if I have to go to, I'm going to Idaho, right, this weekend. What happens if I have to go to the ER there?
Out of network.
Yeah, because I have, Blue Cross Blue Shield of Oklahoma. Not of Idaho. But anyway, this is getting into that realm. But this is why we have this chapter. This is why we're talking about it. There's so much ethics hidden in this chapter. And this is stuff, especially if you're going into any healthcare field at all. And even because you were going into vet, why, talking about insurance for pets or healthcare costs for pets versus even humans or things like this, right? Right. So all of this stuff is stuff to think about when we talk about any of that, even though it might not all be directly related to this, but it gets you thinking about it. Because modern, right? Modern, quote unquote. Isn't it wonderful? So, anyways, moving on. It's probably not gonna be able to cover everything. That's okay. Let's see what I want to say. We already know we can put a gene into this plasmid and then put that into a bacteria and it'll make our product. Bacteria will grow up, crank out that protein, and then we can purify out. There's all sorts of means that we can use to purify out the protein that it's cranking out for us, and then we can give it to you. There is such a thing as synthetic biology. This is creating organisms, yes, organisms or other molecules from scratch. From scratch. I don't mean that we take the DNA from other organisms and paste it together. I mean using nucleotide after nucleotide after nucleotide to design an organism from scratch. I mean, I don't know. Ask these guys because they did it. in 2010. They created a brand new self-replicating living bacterium completely from scratch.
Did they name it Jimothy?
I bet they did.
I hope they did.
I bet they did name it Jimothy. All right, genome analysis. Of course, now that we can look at all these, make all these copies of all these genes and whatever, we can break it down. Basically, we call it shotgun sequencing and try to just hit as many things as we can and then link them up based on their matching up sequences. That helps us build a genome, our understanding of what a genome is. You've heard of the Human Genome Project. That's us trying to map out every possible version of every gene that we know of in all of humankind to understand what's going on with all of us. And so it's doing that. We can also do it using our restriction fragment length polymorphism with our enzymes, restriction enzymes, to look at our criminal or our paternity tests. Okay, that's all that this is talking about. So profiles and maps of everything. We'll put all this into a gel, not unlike what we use in lab, but it's purified version of it. So it's all called agarose. So it's the sugar from agar, nice and pure. And we'll run it through this gel with a nice buffer flowing in it, liquid, and pop like a whole bunch of electricity into it. Because we've talked about what do we have on the backbone of our DNA? Phosphates, yeah, negatively charged. So because they're so negatively charged, they're going to move to the positive end when you do this. So I guess that's what you could say, but that's what we need to know. Negatively charged DNA is going to move to the positive end. So you might be thinking, well, if it just moves through the gel, like how's that going to tell you anything? It's just, okay, it's in a gel, like what? But here's the kicker for it. Certain sizes get stuck. So the bigger you are, the where you're going to get stuck. Not you, but I mean like the DNA. The DNA, the bigger the DNA piece, it's going to be more likely to get stuck. And I mean, it can actually, you could break it down to, I guess, you know, looking at like bouncy balls, going for something like that, sieve of some sort, through different layers. Big, massive balls are not going to pass through as quickly as the little tiny ones that can easily sneak on through. So the smaller, the faster, the bigger, the slower. Small ones go faster, bigger go slower. They just get caught up in the gel. So now we can separate out those different pieces. And you guys have seen pictures of that, I'm sure. You're looking at criminals or whatever, and there's all those little lines on the little chart or whatever. That's that. We separated out their DNA fragments and compared it to somebody else's separated fragments. So this is what it looks like. So you can compare people's little, you know, marks that they got and their markers to see if people are the same. So look at person 3 and look at person 4 here. Probably the same person, huh? Because look at everybody else. You can see the variability here with whatever enzyme or enzymes they used to create this map. So yeah, so that's one way you could say, the blood on, you know, the handle of this, you know, thing, the car that it was reaching for is the same as the blood on the knife that the victim used to defend themselves. It's on your car, so and it's your blood. So that's what they can do. They can say that. We also have something called nucleic acid hybridization. This is taking DNA codes that match one another, complementary to one another, and looking to see if you have the match. So we're using pieces that maybe have fluorescence on them or something that we can look at, and washing over your samples. And then if it sticks, it's a match, a genetic match. That's what this is, nucleic acid hybridization and probes. That's the general concept of how they're going to work. You can use DNA or RNA. You just have to have a known sequence. Let's see. Yeah, so we can do this to see infection from a patient specimen. Look for the matching code if in their swab wound thing to see if it matches staph or something like that, a staph gene, without having to do as much PCR or without having to maybe purify it. You just want to know if it's there. So this is how it would work. It's just a picture of it, and I just want to go to the next thing so that we don't get too stuck on it. Like I said, you can make it fluorescent, and if it is fluorescent and you're going to look at it with an imager of some kind, when you do that, see if it fluoresces or not, that's fluorescent hybridization. And if it is in situ, that's us looking at it in like your actual tissue sample, like or in your actual tissue. So we can look at it that way. So we can look at that. We're not doing this, obviously. I want to click through. I want to get through the stuff. All right, size of DNA, like we said, it's going to be measured in base pairs. K means kilo, just like it always did, right? Kilometer is how many meters? 1,000. So kilo bases, 1,000 base pairs. Okay. So that's how we'll measure our DNA, and there will be questions probably on both of the exams that'll ask you which of the following, piece is going to move faster in a gel. So if it's faster, we want to find a smaller one, and you'll have to be able to tell if I put, 1 base pair versus 200 base pair versus, this many kilobase or this many megabase, which is a million. So FYI. All right, we can create the picture of the genome. We've kind of already seen the mapping aspect of it, but because we can use PCR, we can literally sequence an entire genome just by making sure that our answers are matching up. We already went over these restriction fragment length polymorphisms and what they look like. We already talked about this gel. I don't know why it's on here twice. I feel like it's the same thing again. But yes, you can use it to identify paternity, or you can use it to identify a killer or whatever. So sequence maps, like I said, this is like sequencing a genome to create a map of what kind of gene is wherever. So we can do that with a genome pretty easily with human genome and say, this is a gene for hair color, this is a gene for height, this is a gene for whatever. But it's not going to tell you all the possible versions of that. And so that's what the Human Genome Project is really looking at, is that sort of stuff. But this is how it works. You just do shotgun sequencing with kind of random primers, and whatever you get made out from the end, you can use computers to like match up the sequences, and then eventually with those pieces that you're making, make up a whole genome that way. So that's how it does. That's called bioinformatics and genomics. Cool. Okay, single nucleotide polymorphisms. This is when you have one change in one nucleotide causing an effect in somebody, in their genes. And you can look at, let's say, brown hair versus blonde hair was just one nucleotide change, because that could change phenylalanine to like leucine, just that one change. So that also is what we refer to as a single nucleotide polymorphism. All of the versions that can happen from just one nucleotide change in whatever codon that you want to look at. That's just one codon I'm talking about. So all of the human individual traits that we see in humans versus one nucleotide changes. Human genome has 10 million single nucleotide polymorphisms that can exist, different variations of single nucleotide changing for whatever kinds of genes. That's pretty mind-blowing, considering each of us only has 20,000 genes. Here's an example, thrombophilia. So this is a clotting factor disease. There is a point mutation in one gene, just one nucleotide, I should say. One nucleotide from UUU to UUA, let's say. So phenylalanine to leucine. And that creates that factor, that clotting factor, to be misfolded and cause this disease. Just one. So that's how important those amino acids are. Pretty crazy. This is just talking some more examples of these SNPs, these single nucleotide polymorphisms, and how they can affect the structure. DNA microarray. This is putting a whole bunch of genes, let's say a whole bunch of disease possible genes on here, or different genes of different kinds of streptococcus, or different kinds of salmonella, or whatever. Putting your patient sample on there, mixing it, and then looking for fluorescence if it bound or not. So this is like big scale, like the nucleic acid hybridization. It's sort of like big scale of that. So these can have up to 365 samples on one thing, and they can do, the testing takes like less than an hour for sure. So you can test for all different variations. Pretty cool stuff. So it's microarrays. So we use this for diagnostics. And then I do want to just keep going through this. We already talked about the uses of these guys. You can make some of these things for us, like biologics, to block your inflammatory response. Of course, if you're blocking your inflammatory response is important in your immune system. So that's why people who are on Remicade or Humira or any of these other MABs that are affecting, inflammation usually, are told that you have a decreased immune system. Because you do. It has, your immune system can't react appropriately. It's being muted. Okay. All sorts of stuff you can make. You can even use it for making hepatitis, making vaccines. whatever. GMOs are genetically modified organisms. These are transgenic. These are the same word, GMO and transgenic. We switched up some genes, transgenic, or we have genetically modified organisms, GMO. Anything that has been through recombinant treatment, like we've been talking about, is considered a GMO or a transgenic organism. So it could be large scale, it could be your plants, your corn that's been introduced with this organism, Bacillus thuringiensis, which is a bacterium, and it's a gene from this bacterium that helps them ward off insects, and they put that gene into plants. Almost all the corn that you eat will be that GMO. Guaranteed, you are encountering way more GMOs than you think you are. Way more. You think you pick up the packages and you look at them and you're saved from a GMO. I hate to break it to you. There's a lot more out there. So they can have it like just post on, yep, this is our thing, this is our GMO thing, and post on like their website or something, and they don't have to necessarily make it big and bold on a package, or like on the back of your corn, it'll say it tiny, but they have to say it somewhere, but they don't have to be in your face about it. So the engineer, there's concern about GMOs. I want to put this in the ground before we go to lab. I want to put this in the ground once and for all. If you eat a genetically modified organism, whether it's corn or chicken, which most of you are probably eating GMO chicken also. Yeah, chickens don't normally have that big of a breast meat or thigh meat, like they don't. That's not natural. Do you know how they got that way? GMOs. So yeah, so eating them. So let's say we introduce genes into the chickens and made them that way, and you eat that meat, and your body is going to chew it up and digest it. Your body is also going to chew up and digest everything in it. It's going to digest up the DNA. That means not that you're going to insert its DNA into your genes. It means you're going to break it down to the nucleotides and use the nucleotide. The nucleotides ain't got nothing wrong with them. They're just nucleotides. Nucleotides are nucleotides. So what I'm telling you is, in case you aren't following, when you eat a GMO organism, it does not incorporate itself into your DNA, and it cannot do that. I want to be just end all be all on this.
So all the people that
It
be like,
cannot.
GMO is not good, I don't want to eat.
90% of people are just misinformed about it, right? So this is the issue with the GMO. With plants and things like that, they can transmit their genes because of how like their seeds work and everything like that. They have these special things called transposons that allow them to transmit their genes to specific spots in their genetic makeup. We don't have those spots, but the plants do. So now we're talking about that being an issue. So you might not get it from the plants, but the plants can get it from the plant. So the corn that you're eating that are resistant to pests can transmit potentially that resistance to wild plants. And now you've got a problem, because the whole balance of the ecosystem is off. So that's the big issue with the GMO. Why we have to be sure that we're using, I feel like, of course, using them for a reason and being thoughtful about it, not just making it because we can, right? Because that's a bigger issue. That's what we've been doing. It's just making them because we can. Makes things cheaper in the long run, but not cheaper for them in the long run, but maybe not for the environment. Okay. Oh, okay. Gene therapy. This is the last of it, right? Gene therapy. This is looking for genes that are faulty in humans and replacing them with the right version, the good version, what it should be. So we're talking about actual defects, things where you are not making a protein correctly that could potentially lead to death, or it could lead to deformities or disabilities or lifelong medical care, right? So identifying those genes in an individual gene and giving that person a healthy version of it. The easiest way to do that is going to be by taking the healthy person's gene, putting it into a virus. And we've already learned about how viruses can put their DNA into our DNA. So we can hijack that system. And it won't be the virus itself anymore. It'll just have the genes that we wanted in it. So we can do that. We can choose. We can pick and choose. So we're not giving somebody HIV. We're using the HIV shell to insert good gene into a patient so that they can survive. And that becomes permanent in their cells, but not to their offspring. It doesn't affect your gametes. So it's called what we call somatic cells, is your non-gamete cells. Things that are gonna be heritable or genetic, would have to affect the gametes or the fetus while it's growing, or immediately the baby when it's born. So that's gonna be harder to do, right? Obviously. So we will insert the human DNA into, this is exactly what I'm talking about, into a patient. The best way to do it, let me take your bone marrow out of you. Yeah, that's where all, like most of your white cells are gonna come from. Take some bone marrow out of one of your long bones, swish it up with some designed HIV with the good gene in it. Get those cells infected, and then put it back in your body. And then it can spread out from those stem cells that are in your bone marrow, and make sure that you are covered with that. Get you better. Huh? Yes, I've heard that it hurts too. I don't know enough about it though. I can't donate bone marrow. Okay, so when you can do this, when it's heritable, it's called germline. When it's just in your body cells, it's called somatic. Somatic is not heritable, and this is most of the gene therapy that we're talking about, is the somatic version. It's not heritable. And as of 2022, there were 13 different diseases that could be treated with gene therapy. So we've only got to 13, but they work. Spinal muscular atrophy, inherited vision loss, and acute lymphoblastic anemia, I believe is what it was supposed to say. And others, there are others as well, but these are just some examples. So yeah. We can use RNAs that match a sequence to match it up to DNA sequences to silence it. That makes sense, right? A little bit of RNA, match it up to the gene. It can't work anymore now. It's been silenced. So that's what microRNAs are. And we're not going to go into them too much. I don't ask about them on the exam or anything. But yes, they can interfere with all that. So that's that. We'll go to the lab. I'm sorry for the carryover, but I appreciate the discussion.